And good morning, and thank you to everybody. Yeah, today I will talk about the title reflects the fact that I would like to say you something about genome editing in plants, but something more conventional, as I will show, and something that probably is requires more research in the future. Just a moment. The site-specific nucleases are the basis for all the genome editing approaches in plants as well as in other organisms. Site-specific nucleases, the first kind of that were discovered now 20-25 years ago are meganucleases that are specific enzymes that induce in DNA, and they recognize specific sites in the nucleus, in the DNA. Actually, in the mid 2000, zinc nucleases and TALEN quite a bit later were discovered and set up, and the difference here that I mean, they use a FOC1 as a nuclease to cut the DNA, but the important thing that they have specific sequences. These are the amino acid sequences, protein sequences that allow to recognize, to choose the site, to program to choose the site where to induce the cuts. Later, not much later, but now 10 years ago, more or less, CRISPR-Cas9 was a new method, and this in this case, the cut in the DNA is induced by Cas. But the important thing that the big discovery was the fact that using an RNA molecule, you can choose exactly where to induce cuts. And but anyway, whatever the method, the important thing is that was now discovered at the late beginning of the 90s that were induced a cut in the two strands of DNA. Repair mechanisms are induced, and this mechanism usually, at least in the nuclear genome of plants, are based on non-homologous end joining on the left of this slide here, and on non-homologous end joining repair usually induces random mutation based on short indels, or in case you add a donor DNA, you can have also some gene insertion, or if you have multiple cuts, you have a large deletion. On the other hand, this is much rare in nuclear plant genome. You can induce also have another repair mechanism based on homologous recombination. And in this case, if you add a donor DNA with flanking sequences homologous to the target sequence, you can have either precise gene substitutions or insertion of larger fragments, even genes, in a precise site. Actually, this is an old graph that we published in a review that we wrote some years ago, but just to give you an idea that the research trying to devise new method to have a precise modification of DNA, precise engineering of DNA in plants lasted for quite a long time, but there was clearly a breakthrough in 2013 when CRISPR-Cas appeared after the paper of Jinek, Jennifer Doudna and Manuel Charpentier in the year before. And in a few years, just in 3 years, when we prepared this graph, the number of papers published increased quite a lot due to the impact of CRISPR. And recently, if you can just search the Scopus and the Web of Science, you can retrieve more than 5,000 publications. Actually, after the first version of CRISPR-Cas in 2013, that, as I was saying before, came after zinc finger nucleases and TALEN, there was the development of several CRISPR-Cas-based tools. Several versions of Cas appeared. The Cas molecule was modified in order to have a nickase effect, cutting just one strand or just an activation of nuclease action. And several molecules have been added to the Cas and exploiting the normal mechanism of regeneration based on RNA molecules. Other molecules were added as base editors, transcriptional effectors, fluorescent proteins, epigenetic modulators, and new also methods like prime editors have been developed. And when these molecular tools are delivered into plant cells as a DNA plasmid, RNA molecules, or ribonucleoprotein complexes through tissue culture-based alternative methods, you have several outcomes, and these outcomes can be exploited either in functional studies or in applied breeding. Okay, so based on this, today, as I was telling, I will tell you something of our work on the development of resistance to parasitic plants in tomato using CRISPR-Cas, in editing potato mitochondrial genome using tail-based methods, and also more recent approach, alternative approach based on nanotechnology that we are trying to develop, we and other people in the world trying to overcome bottlenecks of genome editing in plants. As concerns the work in tomato, Felipanche ramosa is the most important parasitic plant in the tomato field, especially in the Mediterranean areas. It's becoming quite a problem, especially where tomato is grown year after year. And seeds of parasitic plants of Felipanche ramosa and other Orobancaceae can remain in the soil for many years until there is the release of strigolactons by compatible host roots, and this release induces germination, the formation of an astorium, and then the colonization eventually of the host root. Strigolactons in plants are synthesized starting from the carotenoid pathway. These are the main genes involved in carotenoid, in strigolacton synthesis in plastids and after in the cytosol. And the final, the main strigolactons in plants, in tomato plants, are those that I reported here, the either orobancol, orobancol, solanacol. So the idea that if you can knock out either one of the genes involved in biosynthesis of strigolactons, you reduce the strigolacton secretion in the soil, and so you reduce the seed germination, the orobanc seed germination. So we designed guide RNA for the main genes involved in strigolacton biosynthesis. We tested the vectors in airy root system, and then we used these vectors in conventional agrobacterium mediated transformation of cotyledon in tomato. The regenerated shoots were subjected to fragment analysis, and then mutant mutations were confirmed by sequences and plants transferred to in vivo in the greenhouse for phenotypization and selfing in order to have the next generation. So this is an example of a mutation retrieved in the different genes, and as expected, based on the approach used, we got several deletions, short deletions, up to 5 nucleotides, and also in some cases, short insertion of just 1 or 2 nucleotides. These plants this is an example with CCD8, CCD7. The data, we have data now also with other genes, but similarly to this, I mean, the plants that had the gene knocked out didn't show any strigolactone in any detectable strigolactone in the root exudates. These imply the less germination in the in vitro germination assay of using root exudates of Felipanche ramosa and Felipanche gypsiaca, that is a related species, not still present in Italy, and not present yet in Italy. And then you can see a reduction in both cases of seed germination. And this was reflected also in a less presence of tubercles. That is the first sign of colonization by Haustorium in the mutant. I mean, especially with the CCD8 mutants, no sign of tubercles was apparent, while in CCD7 there was a strong reduction in comparison with the wild type. When plants grew up, I mean, we could see some effect on the genotype. I mean, strigolactone, as probably know, are involved not only in interaction with parasitic plants, but they have also a role in plant development. And so, as you can see here, mutant plants were shorter, with the shorter internodes, with more higher branching, basal branching. So probably these plants cannot be used as such directly in the field, but we are testing their use as a rootstock, because when you graft a scion on a mutant rootstock, the strigolactones produced in the aerial part of the plant are sufficient to have a normal development. Okay, I mentioned to you before that we used cotyledon explants for agrobacterium-mediated transformation, but also we exploited alternative methods for editing, trying to have a DNA-free editing. We transfected the protoplast with the ribonucleoprotein complexes, and also we tested, we assessed the effect of multiplexing using 2 genes at the same time. In this case, we used two guide RNA for each gene, and we transfected with the 4 guide RNA at the same time, complexed with the Cas. And the different guides have different efficiency. In most cases, they were induced the short deletion insertion at the site, but also in some cases there were long deletion, as you can see here as an example in the CCD8 gene, expected 193-94 nucleotides between the 2 guide RNA sites. Overall, so we could demonstrate in the calli, unfortunately, because we were not able at that time to regenerate shoots from calli, but so we assessed the efficiency of editing at the calli stage And there was a very high frequency mutagenesis, also high frequency of multiple mutation. And because we didn't use any plasmid, we didn't have any integration. So this is just a transient effect of the complex transferred in the cell into the protoplast cell. Anyway, we made a step forward in collaboration with some colleagues at Sweden and also with the Chinese researcher. Later we obtained the regeneration in a few genotypes of tomato, and so this is an advancement, but I mean, as you probably are aware of, protoplast culture is a laborious method and also is strongly dependent on the genotype. Okay, now this is the work on tomato. Now I will tell you something about the work on editing potato mitochondrial genome using TALE methods. I mean, just to, as a reminder, plant mitochondrial genomes can interact with the nuclear or directly can affect the performance of natural population crops. And cytoplasmic male sterility is one of the most known traits. Crop chondrioms are quite different plant chondrioms in comparison with the animal ones, especially human ones are very big, and there is a large variability in between crop plants. As you can see here, there is the presence of subgenomes that derived from recombination of numerous repeats, and the presence of different kinds of molecules. And so on the other hand, compared to the variability at the structural level, there is a slow mutation rate at the nucleotide level. The genetics of plant organelles, mitochondria and plasmids, is not easy, and is the limited information based on conventional genetic approach due to the absence of genetic recombination and uniparental transmission after sexual cross, usually through the maternal parent. On the other hand, there is a quite a good frequency of recombination rearrangements of parental genomes after somatic hybridization, but the possibility to get this is depends on the availability of a protoplast system in plants, and also the analysis of this variation is quite long and not easy. Transformation methods are not available for higher plant mitochondria, so this reduces the possibility of easier straightforward analysis of mitochondrial genes. And also we use the tail-based method approach because CRISPR-Cas approach are not readily usable in mitochondria and in plastic due to the difficulty to import the Cas and especially the guide RNA through the double membrane of plant organelles. So the strategies we used are based on two approaches. One is the mitoTALEN. In the case of mitoTALEN, the tail sequences are fused with the nuclease FOC, as I told you, and FOC1 functions as a nuclease one is a dimer, so the two subunits are transferred into the nucleus through agrobacterium transformation with a regular normal binary vector, and then through a mitochondrial localization signal is transferred to the mitochondria where the double-strand break is induced. And this, in contrast with the nuclear genome, in mitochondria, usually repair of the double-strand break is through the homologous recombination pathway, not from the non-homologous end joining. In the alternative approach that we used, it's a base editing method in which cytidine deaminase that is derived from the bacteria that is able to work on double-stranded DNA is like in the former case is transferred to the nucleus through agrobacterium transformation, and then is localized through a mitochondrial localization signal in the mitochondria, and then the objective is to convert the target C in a thymine and in a target site in order to induce a stop codon. So this is what we did in using a potato somatic hybrid and specifically ORF, a gene, short gene, 378 base pair that is an ORF that is present, or ORF 1 to 5 that is present in Solanum tuberosum, but not in other Solanum species. And we are interested in assessing and understanding its function. So we, on the left, we designed two tail sequences in two sites that are the yellow one, and in between them underline is the site where the FOC1 induces the cut. On the right, you can see in yellow violet the two sites for two tail sequence pairs, and in red is the C that we hope to convert in T in order to induce premature stop codon TAA in place of the CAA shown here. So these are the results first of the TALEN. The bottom is the target region. The ORF 1 to 5 is in between the two genes, one very important, the NAD4, the first two exons of the NAD4, and then upstream ORF 1 to 5, there is another ORF 247 of unknown function. And so at first we amplified the total DNA of transgenic plants using primer landing, P3 primer landing on the first exon on NAD4 and P6 that is a primer in between in intergenic region between the ORF 1 to 5 and ORF 247. And also we had two internal primers for the target gene. And as you can see here in the experiment with the first primer pair, the first tail sequences T1, 3 plants using P3, P6 primers, we expected a fragment of about 1,000 base pairs that was obtained in the majority of plants except in 3. In T1, 60, 29, 49 plants, as you can see here, there was no amplification. And the same was confirmed below with the internal primer. On the other hand, using the second pair of T2 in another experiment, in 2 plants, 12 and 14, we had the same result, no amplification. In other 6 plants, we had the appearance of a fragment, a new fragment, indicated with the red arrow, and this fragment was about 200 base pair shorter than the expected one. In 2 plants, 10 and 31, this fragment was the new fragment was the only one present, indicating homoplasmy. In other 4 plants, 1, 23, 26, and 29, there were both fragments indicating a situation of heteroplasmy. So as a first, we trying to understand what was the origin of this new fragment of the T2 plants. So here we sequenced this fragment, and we could see that it was 747 base pair long, so 236 base pair shorter than the expected one on the left. And also we could see that in the original sequence, there were 2 direct repeats in red, that after FOC1 induces the cut in between the tail sequences, are responsible for repairing, and then in the repairing process, there is the deletion of the sequence in between them that is exactly 236 base pair. So this recombination, homologous recombination between the two direct repeats here induces the repair and the deletion of the intervening sequence. Besides that, we checked the target gene, all the regenerated plants, for the presence of additional mutation using fragment restriction analysis. And in only one case, in the plants 26, we discovered that the plants 26 actually is quite interesting because as the mutation, the 236 mutation, the deletion, and also has an additional mutation in the normal fragment that is an insertion of 4 base pair. So has 2 mutations in the same plants. Then we switched to the other 5 plants that 3 in the experiment 1 and 2 in the experiment 2 that just remind you that are derived from using 2 tail sequences in 2 sites. And in this case, these plants didn't show any amplification with the P3P6 primer pair. So we tested first with the presence of the 2 genes at the boundaries of this region. And as expected, they were present, especially, the DAN4 gene. We didn't expect that the plant could grow without it. So it was present in all plants, but also the other half, the 247, was present in all plants. So the 2 flanking genes were there. So we tested, we used the primer upstream, P7 upstream of P6. And in this case, we got a fragment and amplification in 4 plants of 5, not in T212. And in these 4 plants, the new fragment with the red arrow was about 850 base pair long, instead of the expected 1,919 in the original SHN9B. So looking at the sequence again, we had the proof that the fragment, the mutant fragment, is 853 base pair long. So it was 1,066 base pair shorter than the expected one, the original one. And again, we could find 2 sequences here, 2 direct repeats, different from the previous one, that are outside of the region, but close to the region where the 2 T1 and T2 tail sequences induce the cut. And so again, through the repair of these cuts, there is a deletion that in this case is 1,066 base pair long. And so again, it's confirmed that the main mechanisms of repair in mitochondrial genomes is homologous recombination of direct repeats. In this case, direct repeats are 11 and 12 base pair long, so quite short. So the last plant that remained to understand what happened was the T212. T212 that I remind you didn't give amplification with P6 and P7 primers, and again didn't give any amplification with upstream primer P8 and P9, but it gave an amplification, gave a fragment, an amplicon of about 800 base pair using the P3 and P11 primer that land of the 2 genes. And in this case, so probably the fragment is 800 base pair long instead of 5,100. So probably a deletion larger, the longer deletion of 4,300 base pair long was obtained. We are looking at sequences. We have preliminary analysis results. We don't have yet the clue, I mean, what was the origin of this deletion, what were the molecular mechanisms involved. But it seems quite clear that this deletion was probably derived by similar mechanisms to other deletion shown before. So this is the synthesis of the results of mitoTALEN. With the first tail pair, we had 3 edited plants out of 15 regenerated transgenic plants, and all the 3 show the deletion 1066 base pair. In the second experiment with the primary pair, 8 plants out of 14 were edited. 6 show the short deletion of 236, 1 the deletion 1066, and another one probably 4,300. Okay, so this is a graphical representation of the 2 recombination that we could, mechanisms that we could evidence, and as a result of that, we have a shorter protein, or we have just no protein at all. In this case of the deletion with the 2 sequence here, the yellow one. Concerning the base editing approach, we also in this case, remind you, we used 2 sites. These are the violet are the tail sequence, and this is the window between them, and C is the target C for conversion to T in order to induce the stop codon. And clearly this it seems that overall there was this conversion was 12 plants out of 21 in the first case, and 10 plants after 22 in the second one. But in the second, with the second tail, in the second site, the efficiency was generally higher, because there were most of these conversions were in homoplasmic stage. Instead, in the first case, there were most of the plants were heteroplasmic. Anyway, these plants were gave origin to shorter protein. Expected that there should be 56 amino acids or 88 instead of 125. And also, but besides this, we also got some other missense mutation in other C present either in the same strand or in the opposite strand in the window. And we are now trying to understand what is the effect of this missense mutation. And but no mutation were obtained, not off target mutation were obtained out of this target window that is 15 nucleotides. Okay, just in the last few minutes, I would like to tell you something about what we are doing, trying to develop and implement in crop plants new methods for editing, trying to avoid the novo regeneration transformation in vitro, because the ability to regenerate and being transformed in plants is a bottleneck for genome editing. Is the effect of genotype is strong not only at the species level, but also in species that respond. There is a strong effect of between different genotypes, and so this is a strong limitation for applying genome editing, especially by seed companies and so on. And then also must be said that the frequency, even the best cases, is sufficient. The efficiency for gene knockout based on non-homologous end joining, that's generally too low for application based on homologous recombination. Besides that, agrobacterium mediated and other transformation approach usually induces integration of the Cas as a gRNA cassette. That is not a good thing. Then you should get rid of that by selfing or crossing where it is possible. That is more difficult in vegetatively propagated species. Then you have to use induce recombination, and also it is good to use transient method to induce editing. That is, we show that in our case, but also in other species, we use in tomato ribonucleoprotein complexes. That is efficient method, but should regeneration from protoplast is generally laborious and strongly dependent on genotype. And then there is also the effect of somaclonal variation. That is probably a problem. So in several labs in the world, there is a strong interest in developing tissue culture independent genome editing approach. One that works quite well is floral dip, but unfortunately it works well in Arabidopsis, just in Arabidopsis and few related species. Other methods are being developed in other species as using editing, inducing editing in haploid inducer, in zygotes in vitro, or trying to target meristem or induce de novo induction in vivo in meristem, or using viruses, and also nanoparticles that actually, but both nanoparticles, as you are aware of, nanoparticles, there are different types of nanoparticles. They can be used as to deliver DNA, but also other kind of molecules, RNA, ribonucleoprotein complexes, at least in animal cells. The problem of plant cell that they have the cell wall that is much more difficult to cross, and so deliver molecules into the cell. But there are several attempts to use nanoparticles, different kinds in different cells or organs, and the aim is to transfer the editing reagents into the cell. We have started quite recently working on that. We have some preliminary but interesting results with metallic magnetic particles, either charged positively for DNA binding or negatively. We use this as a reporter system with the fluorochrome. We are carrying out some different experiments to bind DNA. We are looking at efficiency. Clearly, we need to improve the efficiency of binding, but we could demonstrate, and we are using pollen as a first attempt. The aim is to edit pollen and then use the edited pollen to cross, to pollinate plants. And this pollen was not affected by camera particles, so the germination was even improved, as sometimes happens with nanoparticles that have a positive biological effect. And also we could demonstrate that using the fluorescent particles, and you can see at the regular microscope, there was a strong binding of the outer surface of the pollen grains, and by confocal microscope we could also demonstrate a preliminary indication that there is also internalization of some particles within the pollen grains. But I should say that this is probably a long way to go, and there are two main limitations nanotechnology-based genome editing in plants. One is the delivery of large Cas molecules through the cell wall. And but you are probably aware of that new Cas molecules have been discovered recently, not only in other bacteria but also in phages, and these more compact molecules are probably would be interesting to see their efficiency and the possibility to deliver them to the using nanoparticles through the cell wall. The second aspect that is also a limitation, that in order to transfer the induced mutation to the progeny, you have to target the germline. And also this is not a trivial task. Okay, so finally, I would like to thank all people with whom I interact and with whom I collaborate on the three topics. The editing of tomato plants for resistant to parasitic plants is a work mainly carried out at the Research Center for Vegetable and Ornamental Crops in Ponte Cagnano, and is coordinated by Alessandro Nicolia, that coordinates not only a group of people out there, but also other people, Nunzio D'Agostino University of Naples, other colleagues at the CNR Institute in Bari and in Avellino, and people at the University of Alnarp in Sweden and Heime University in Japan. The work on potato mitochondrial genome is a long-term collaboration with the Nunzia Scotti here in Portici, with the precious technical assistance of Lorenza Sannino, and also in this case there is a collaboration with Nunzio and colleagues, other colleagues at the University of Naples, with Alessandro Nicolia at the Ponte Cagnano, and Shinji Rimura, that actually developed the mitotalent base editing approach in plant in University of Tokyo, with whom we are collaborating strictly. And the new approach based on nanotechnologies, I collaborate and closely collaborate with Alessandra Ruggero in Georgia, Batelli in Portici, with Alfredo Ambrosone, Lisa Cappetta in Salerno, Alessandro again in Ponte Cagnano, and Maria Moros at the Universidad de Zaragoza in Spain. And I would like to thank you for your attention.